Hello everyone, welcome to INI-CET November 2023 discussion of biochemistry. I'm Dr. Rebecca James, your faculty with Marrow. Most of the questions in biochemistry were very simple question, straightforward, one-liner type of questions. One or two question was a bit tricky. Apart from that, everything was simple questions only. So we'll dive into the question. The question number 1. 2,3-BPG binds to heme part of myoglobin, globin part of hemoglobin, globin part of myoglobin, heme part of hemoglobin. So here the 2,3-BPG binds to the globin part of hemoglobin. That is the answer. What about heme part of myoglobin and the heme part of hemoglobin? Oxygen is the one which is binding to the heme part of myoglobin as well as to the hemoglobin. So what about the globin part of myoglobin? Does it have any interaction with the 2,3-BPG? No, globin part of myoglobin has no interaction with the 2,3-BPG. Now what about this 2,3-BPG? This 2,3-BPG is synthesized by RBC, where 10% of the RBC that entering into the glycolytic pathway is entering into a shunt pathway which is called as the 2,3-BPG shunt, otherwise called as Rappaport-Lubering shunt pathway, or simply the RL shunt. Which enzyme is bypassed when it is entering into that RL shunt? It is 1,3-BPG bisphosphoglycerate kinase. This is the enzyme that is being shunted when it is entering into the RL shunt. Now, what is the role of this 2,3-BPG? This 2,3-BPG is it can bind to hemoglobin. How many 2,3-BPG can bind? Only one 2,3-BPG can bind to one hemoglobin. Then what about oxygen? Oxygen, 4 oxygen is binding to one hemoglobin, because we know that there are 4 globin chains. So we see the structure of hemoglobin, where we can see 4 globin chain, and each of this globin chain is attached to a heme. So each of this is heme, and each of the heme is binding to one oxygen molecule, which means that hemoglobin can bind to 4 oxygen molecule. Now, where is this 2,3-BPG binding? The 2,3-BPG is binding to the central cavity, which is formed by the 4 globin chains. Okay, so in the central cavity, only one 2,3-BPG can bind, not 4 2,3-BPG. Now we will see a magnified view of the central cavity. So if we see the central cavity, what you can see is this is a valine amino acid which is present in the beta-1. So you can see this is 2,3-BPG. Now this 2,3-BPG, what is the charge of this 2,3-BPG? It's negatively charged. Okay. Now, this negatively charged 2,3-BPG is attached with the positively charged amino acids and positively charged group of the amino acid present in the globin chains. Which globin chains? These are the beta globin chains. Okay, I'll explain that. So, what you can see is this is the beta-1. This is again a beta-1, beta-1 and beta-1. So, apart from that, you can also see beta-2 globin chain as well, which means this 2,3-BPG, which is seen in the center of this cavity, is forming a bridge. What is that bridge? This bridge is called as salt bridges, otherwise called as the ionic bridges. So it forms ionic bridges or salt bridges with certain positively charged amino acids and positively charged groups present in the amino acid, and that too only to the beta globin chains. So even if it is present in the central cavity formed by the 2 alpha and the 2 beta chains, the salt bridges are seen only seen attached to the beta globin chain. So this is one thing that we should understand. So in short, what we have to learn is the 2,3-BPG is seen in the central cavity formed by the interface of the 4 globin chain, but the salt bridges are formed with the terminal amino group of valine and certain other amino acids like lysine and histidine, which are present in the beta-1 and the beta globin chains, which are designated as beta-1 and beta-2. Okay, so what is the importance of this lysine and histidine? They are basic amino acid, so naturally they are positively charged. Apart from that, in valine it is attached to the amino terminal. So this amino group, we know it is positively charged. Okay, so that is about the attachment of 2,3-BPG to the hemoglobin. So now we are coming to the function of this 2,3-BPG in hemoglobin. Why do you see 2,3-BPG attached to this hemoglobin? Because when this 2,3-BPG is bound to the hemoglobin, this hemoglobin is forming bridges, salt bridges with 2,3-BPG, which means that the hemoglobin is in a taut state. Taut state means what? Deoxygenated state. So in the taut state or in the deoxygenated state, it has less affinity towards oxygen, which means the 2,3-BPG decreases the affinity of hemoglobin towards oxygen. So in other words, if you are taking a sponge and as a hemoglobin, what you can understand from the taut state is like if you shrink the sponge like this, then it squeeze all the water out. So just like that, it squeeze all the oxygen out. It has no affinity towards the oxygen. So this is called as the taut stage, and 2,3-BPG stabilizes the taut stage of hemoglobin. Then why this is happening? It is because it is a compensatory mechanism that is happening when there is excess carbon dioxide which causes the acidosis. So this is a compensatory mechanism in the peripheral tissues when carbon dioxide induced decreased blood pH occur. So what happens in gamma chain in hemoglobin F? So in hemoglobin F, it has less affinity towards 2,3-BPG. So what is the peculiarity of the fetal hemoglobin? The fetal hemoglobin always want to extract oxygen from the maternal side, which means it should be having a high affinity towards oxygen, which means it should be in a relaxed state so that it can accept or breathe in maximum oxygen into it. This 2,3-BPG has very less interaction with the hemoglobin F. Why? It's because HbF has 2 alpha and 2 gamma chains. So in this gamma chains, what you can find is instead of these positively charged amino acids, what you can find is amino acids like serine. So the serine, we know that it is a neutral amino acid which doesn't have that positive, it cannot form a positive core, which means it cannot attract the negatively charged 2,3-BPG. So that is what is written here. The 2,3-BPG binding pockets are replaced by serine. So the interaction of the hemoglobin towards 2,3-BPG, HbF towards 2,3-BPG is less, which means it always keep the hemoglobin in a relaxed state to breathe in more oxygen towards it. Hope you got it. So in general, this is a very commonly asked question. So we'll just go through this oxygen dissociation curve and the factors which increases as well as decreases the affinity of hemoglobin towards oxygen. So what we have already seen is the oxygen dissociation curve shift to the right means that it is decreasing the oxygen affinity. So what are the factors? Number one, we know acidosis. Acidosis is also written as increased H plus ion concentration, increased PCO2, then increased 2,3-BPG because it facilitates. All these are favoring the T state, okay? Then hypothermia, that is if temperature is high, then sulfhemoglobin, it is towards the right, and also chronic anemias. So all these things will decreases the oxygen affinity and shift the oxygen dissociation curve towards right. Now what about the left shift? Almost whatever is opposite to what we have discussed now is the shift of oxygen towards left, which means it is increased oxygen affinity. So that is, one is alkalosis, then decreased PCO2 and decreased 2,3-BPG, hypothermia. These are the most important one. Then if you want to remember, you can also remember about methemoglobin, carboxyhemoglobin and fetal hemoglobin. Why fetal hemoglobin? Because it has less interaction with the 2,3-BPG. Okay, so now we are moving on to the next question. So the next question is cytochrome P450 monooxygenase utilizes which of the following cofactor during the phase 1 detoxification reaction of xenobiotics? So here we all know cytochrome P450 is a monooxygenase which uses NADPH. So this is the overall reaction that is catalyzed by cytochrome P450 as it is given in Harper, that we can see that the NADPH is the coenzyme here. So what you can see is this is a drug that we are taking. So this is the molecular oxygen that is being added. Now when this reaction is occurring, you can find that one oxygen, only one atom of oxygen is added to the drug so that it becomes ROH, that is, it is getting hydroxylated. So this reaction is a hydroxylation reaction. So few points to remember about cytochrome P450. One is, this is a major component that is involved in the phase 1 xenobiotic reaction, that is hydroxylation reaction. The second is, it is a monooxygenase. The other name of monooxygenase is mixed function oxidase. Then we can see it is a hemoprotein because we know cytochromes, it contains heme. So that is again another point, that is cytochromes. Almost all cytochromes contain heme. So it is a hemoprotein. Then another thing is like the hydroxylation. What happens, or why do we need hydroxylation reaction? That is major, it is to metabolize the xenobiotics. Xenobiotics means the foreign compounds, which includes our drugs. So more than 50% of the drugs are metabolized by the cytochrome P450 system, and for that we need NADPH. So that is what is written. Then the highest amount of cytochrome P450, where do you find? It is found in the liver cells. Not only in the liver cells, also in the enterocytes. So these are the two sites where cytochrome P450 is found maximum. And which is the organelle where this reaction is occurring? It is the microsome. What do you mean by microsomes? Microsomes is the smooth endoplasmic reticulum. Apart from that, it also takes place in the adrenal gland. In the adrenal gland, the major site is mitochondria. Few endoplasmic reticulum reactions are also there, but mitochondrial reaction, this cytochrome P450 mediated hydroxylation reaction, takes place in the mitochondria as well. So going to the next question, which of the following is complementary to the active site of the enzyme? The answer is a clear-cut answer, that is substrate. But there are some students who overthink about some other theories in the binding of the enzyme towards a substrate. We know there are three theories. One is the Fisher's template theory. The second is the William-Jenck and Linus Pauling. The third one is the Koch-Lenz induced fit theory. Induced fit. So, out of these three theories, we know Fisher's template theory is the one which is saying that the substrate and the active site of the enzyme and the substrate are complementary to each other. So, what about William-Jenck? The William-Jenck is saying that the active site of the enzyme is complementary to the transition state, so that maximum products are produced. Then Koch-Lenz induced fit theory is saying that it is just like wearing a gloves, where there is no complementarity between the substrate and the active site, but when the substrate binds to the active site, it induces some changes in the active site so that it is fitting into the this active site. So these are the three theories. Then how we will go about this? So if you have some doubts like this, I should make this point clear. Unless and until a specific theory's name is not mentioned. For example, in this case, it is not mentioned whether it is Fisher's or Koch-Lenz or William-Jenck Linus Pauling theory. So if it is not mentioned, you have to take the common thing, that is the Fisher's template theory, that is, it is complementary to the substrate. So if specifically mentioned that William-Jenck and Linus Pauling, then we can say the answer as transition state. Otherwise, you should not give the answer as transition state. Okay, so this is a picture where it is clearly mentioned that the active site or the catalytic site is complementary to the substrate. Now, what is this site called as? This site is called as the allosteric site. And who binds to the allosteric site? The allosteric effector, otherwise called as the allosteric modifier. So, this is the picture based on that question, that is the active site and the substrate are complementary to each other. Now, going to the next question. Glycogen phosphorylase require which one of the following for its activation? So, the first is insulin. You know, glycogen phosphorylase is an enzyme in glycogenolysis. So, what is the role of glycogenolysis? To push the glucose into the blood when the body is fasting. So, definitely it is not insulin. Then glucose 6-phosphate. We know that when the glucose is pushed from the glucose, from the glycogen, this glycogen is first, the glucose is first released as glucose 6-phosphate, which has to be converted to glucose. This happens in the liver. Okay. Now, in the muscle, this step is not there in muscle. Okay, in muscle it is not there, but liver it is there. Okay, whatever it may be, glucose 6-phosphate is there as a product of glycogenolysis. So, it is never an activator. So, that is also out of the question. Then we have two options here, calcium and cyclic AMP independent protein kinase. It is not actually cyclic AMP independent protein kinase, it is cyclic AMP dependent protein kinase. Cyclic AMP independent kinase is there, which I will tell you, I will elaborate it with a diagram. So, this is not cyclic AMP independent, it is cyclic AMP dependent. So, if that independent is there, that is also not the answer. Then here one possible answer is calcium. So, I will explain it with a diagram. That is, we know that in glycogenolysis mechanism, that is regulation of glycogenolysis, it is done by two major, the hormones that is involved here is the glucagon, epinephrine and also insulin. So, we will see the action of glucagon. We know that glucagon bind to a receptor which is called as G protein coupled receptor, as well as you can see that this epinephrine is also binding to the G protein coupled receptor, but the difference is glucagon acts in the liver, but then the epinephrine, you can see that it acts both in the muscle as well as in the liver. Okay. So, when this is acting, we know that this ATP is converted to cyclic AMP by the enzyme adenylyl cyclase. So, this cyclic AMP in turn activates an enzyme called as cyclic AMP dependent protein kinase, which in turn activates another kinase. This is called as glycogen phosphorylase kinase, which can in turn phosphorylate another enzyme. So, what is that another enzyme? Glycogen phosphorylase. So, this converts glycogen phosphorylase from the inactive state to the active state so that glycogen is degraded. So, if glucagon or epinephrine is binding to the GPCR, what we see is glycogenolysis is taking place. Now, when this glycogenolysis is taking place, the cyclic AMP is acting or this activating which enzyme? Cyclic AMP dependent protein kinase. Okay, it is not cyclic AMP independent protein kinase. Now, what is the role of the cyclic AMP independent mechanism? There, the role is by calcium. What is the role of calcium here? During, this is mainly in the muscle, during muscle contraction, this from the sarcoplasmic reticulum, what we can see that the calcium is released. Now, 4 calcium can bind to another protein which is called as calmodulin. So, once the calmodulin binds with the calcium, this is called as calcium calmodulin complex. Now, what's the action of this calcium calmodulin complex? So, once the calcium is out of the sarcoplasmic reticulum in the muscle, calcium calmodulin complex is active. So, you can see this is active. Once this is active, it is not acting on cyclic AMP dependent protein kinase. Cyclic AMP dependent protein kinase is exclusively dependent on cyclic AMP. It is not under the control of calcium or the calcium calmodulin complex. But this calcium calmodulin complex can bind to this glycogen phosphorylase kinase, which is a cyclic AMP independent. It is not cyclic AMP dependent, which in turn converts glycogen phosphorylase to the active form, which means calcium can activate glycogenolysis. Not a cyclic AMP independent protein kinase. Cyclic AMP dependent protein kinase also can activate glycogenolysis, or in other words, glycogen phosphorylase. I hope that is clear. Apart from that, we should also understand there is another molecule which is called as adenosine monophosphate, that is AMP. This AMP can activate this glycogen phosphorylase without being it phosphorylated. See, all these are kinases, which means it is phosphorylating glycogen phosphorylase. But AMP is a different mechanism where it can convert the glycogen phosphorylase to active form without being phosphorylating this glycogen phosphorylase. Okay, so at what or in what stages these AMP is acting? That is in extreme conditions where there is deficient oxygen, or that is called anoxic state. So one extreme conditions like anoxia or depletion of ATP. So this is the time where the AMP is directly pushing the cyclic glycogen phosphorylase to active state. Okay. So these are the different mechanism. Then about the allosteric. So what we just see is the covalent modification which is done by the glucagon, epinephrine. And what is the action of insulin? I forgot to tell you that. The insulin is like it activates an enzyme called as phosphatase. Phosphatase means what? Remove the phosphate group. So this insulin is like the insulin convert the active form of glycogen phosphorylase to the inactive form. So, insulin is never an activator, it is an inactivator. Okay. Now, going to the allosteric regulation, where you can see glucose 6-phosphate there. You can see the glycogen phosphorylase in the muscle as well as in the liver. So, glucose 6-phosphate is an inhibitor. Here also glucose 6-phosphate is an inhibitor. ATP is also an inhibitor. Then glucose is an inhibitor where in the liver, not in the muscle. So in the liver we cannot, in the muscle we cannot find glucose. Why? Because glucose is not a product of glycogenolysis in the muscle. Okay. So from this diagram also we understand that the glucose 6-phosphate that we have mentioned there is a inactivator of glycogen phosphorylase. That is by allosteric mechanism. So we'll just go to that question once again. Glycogen phosphorylase requires which of the following for its activation? Insulin, no. Glucose 6-phosphate, no. Cyclic AMP independent, no. But the answer here is calcium. Now going to the next question. This is one of the most difficult question I find in the INI-CET 2023 session. That is, which of the following is not involved in the repair of the DNA? So the answer here is MSH2. That is the answer. Why it is the answer? I will explain. There are various DNA repair mechanisms and various genes and its products, that is called as the proteins, are involved in the DNA repair mechanism. So about nucleotide excision repair, there are few genes that I have mentioned. There is XPA, XPC, XPD, etc. If you find it difficult to learn, you just learn that this nucleotide excision repair, there is a disorder associated with it. What is that disorder? It is xeroderma pigmentosa. So that disorder has X and P in it. So whatever with XP is involved in nucleotide excision repair. Apart from that, there is one more complicated name there, that is ERCC1, that is excision repair complementation group 1. This is a group of proteins where it is also involved in the nucleotide excision repair. If you forget it, okay, you don't have to learn everything, but at least learn XP. So whatever starts with XP is with nucleotide excision repair. Clear? Now go to the mismatch repair. There are various mismatch repair genes. The two very important ones are the MSH2 and MLH1, MSH2 and MLH1. So you can see here that there is MSH2 here. MSH2 is involved in which repair mechanism? Mismatch repair mechanism, not with double strand break repair mechanism. Hence, that is the answer. We will see the other option as well. Base excision repair. Base excision repair is a mechanism that happens when there is a mutation in a single basis. Okay, so for that, what we do is first we excise or remove the base from the nucleotide. So to remove the base from the nucleotide, the enzyme that is required here is the DNA glycosylase. Okay, so the genes or the proteins. So here it is the protein, that enzyme, DNA glycosylase, and you can see that another protein that is involved here is MUT-YH. So MUT-YH is the human homologue of E. coli MUT-Y gene. So that is involved in base excision repair. So this is very easy to remember because base excision repair mechanism, what is the disorder that we have learned? MUT-YH associated polyposis. So from that you can remember this MUT-YH there. Apart from that, flap endonucleases. Then just like ERCC1, here there is X-ray repair complementing defective repair 1. So it is very complicated. I know that. XRCC1. Then DNA polymerase beta, that is almost involved in every repair mechanism. We cannot say it is only for this base excision repair. It is there for everyone, but I have just mentioned it here, that's all. DNA ligase, of course, all the polymerases and ligases are involved in almost every repair mechanism. Then one protein that is very specially involved in base excision repair is the PARP. PARP is a protein, it is an enzyme that that is doing one process which is called as ADP ribosylation. ADP ribosylation is associated with DNA repair. Okay, so I have mentioned lots. We cannot see any of these genes or proteins in the option that is given here. So that is also not seen. So base excision repair, at least remember MUT-YH. Then polymerases ligases is there for all repair mechanism. Then also remember this glycosylases. It is exclusively for base excision repair. Then one more which I have given here is the XRCC1. If you can remember, okay, otherwise also fine. Okay, now go to the double strand break repair mechanism. Double strand break repair mechanism has two mechanisms under that, which is one is the homologous recombination, the other is the non-homologous end joining. Now, first about homologous recombination, we have seen many disorders associated with the homologous recombination. One is the Brca1 and Brca2 defect associated disorders that we have seen. So Brca1 and Brca2 is a protein and corresponding gene is involved in the homologous recombination. Then there is a complex. That complex is called mRN complex. What's the role of this mRN complex? That is to sense the DNA damage. So DNA damages, if it is there, this trimolecular complex is sensing the DNA damage. What is this mRN stands for? mRN stands for one protein involved or in that trimolecular complex is MRE11 and the other one. So M stands for MRE. Then R, you can see the R stands for RAD50 and there is one N, that N stands for NBS1. This is called mRN complex which has MRE, RAD50 and NBS1. This trimolecular complex help in the homologous recombination. So the next mechanism is non-homologous end joining. In non-homologous end joining, where a homologous chromosome is not involved there, what happens is that if there is a double strand break defect here, so if this is a double strand break, what happens is this will joining, end-to-end joining of these chromosomes, that is what, or these DNAs. So this is a DNA where there is a double strand break. So these two DNAs are joined or tied together like an end-to-end joining. So this is called non-homologous end joining. In non-homologous end joining, certain proteins involved is DNA protein kinase. DNA ligase, I said, it is there for almost every repair mechanism. But specifically for non-homologous end joining, this DNA PK, that is DNA protein kinase, and Cu proteins. Two Cu proteins are there, Cu70 and Cu86. So you can see here Cu70 is given in this option. Then another endonuclease. This endonuclease which is involved in non-homologous end joining is called Artemis. It is a structure specific endonuclease. So just learn that this is another endonuclease involved in non-homologous end joining. At least learn these two enzymes, that is DNA protein kinase and Cu protein, which is involved in non-homologous end joining. Now we see the options here. Brca1, where is it involved? It is in a double strand break repair mechanism. Then Cu70, you can see it is also a double strand break repair mechanism. Now what is the role of this ATM? So even if you don't know about the ATM, you know that ataxia telangiectasia Ataxia telangiectasia is a disorder associated with which repair mechanism? Homologous recombination. So this ATM stands for ataxia telangiectasia mutated, which means it has some role in double strand break repair mechanism. So in that way you can exclude that option. So in this case, in this kind of questions, definitely you may not be knowing the answer correctly, but you can exclude few options like ATM. Definitely we know it is double strand break repair defect associated thing. So Brca1 also we have learned like that. Cu may or may not, I don't know whether you know it. At least you can exclude two options. Then you are left with the two options, then you can make a guess on that, okay? So this is how you can approach such questions if you are totally unaware of the name of these proteins. Okay, now a few words about this ataxia telangiectasia mutated. That is, even though I have told you many things about this double strand break repair, I have never involved that protein called ATM here in the gene. So the proteins involved in the double strand break repair mechanism. Why? Because ataxia telangiectasia mutated, that is that gene and the corresponding protein, the role is not direct a repair. It is like the important thing is that it is the DNA damage response. Just like the mRNA complex I have told you, this ATM is actually initiator of this mRNA complex. So ATM is an initiator of mRNA complex. So it is not directly involved in the repair mechanism, but it is an activator of this a complex which sends the DNA damage. So this is so that the key point there is it is a initiator of DNA damage response. Right. Then the next point is how it acts. So it is like a kinase activity. Ataxia telangiectasia mutated that active, if it is active, then it uses a kinase activity. So that this is essential not only for DNA repair but also for cell cycle arrest, apoptosis, cell cycle checkpoints, etc. So ATM has multiple role where it is involved in all these activities, not only the DNA double strand break repair, but it is also involved in double strand break repair. That's all. Then, so this ATM is usually considered as a tumor suppressor. So the gene is considered as a tumor suppressor gene where it always helps the DNA to activate this mRNA complex and sends the DNA damages. Okay. Then another thing is that this is ATM. Okay. So this ATM activates Brca1. So what's the role of Brca1? The Brca1 is involved in the double strand break repair mechanism by homologous recombination. Apart from that, we know ATM activates mRNA complex. ATM activates Brca1. All these are involved in double strand DNA break repair mechanism, that is HR. Okay, so this ataxia-telangiectasia mutated, that is ATM gene mutations are associated with ataxia-telangiectasia. Apart from that, so that is also associated with homologous recombination, plus various cancers are associated with this particular gene. So what is the answer here? The answer here is MSH2, which is involved in mismatch repair, ATM is involved in double strand, BRCA1 is involved in double strand, Cu protein, that is Cu70, is also involved in double strand break repair mechanism. So that is giving you the answer MSH2. Now going to the next question, which biochemical cycle or process is used by monoclonal antibody in hybridoma technology? So the answer here is purine salvage pathway, TCA glycolytic pentose phosphate pathway. No. So what is the role of this purine salvage pathway in hybridoma technique? So in hybridoma technique, it is a technique that we are using to develop or produce monoclonal antibody. Okay. Now it's a it's an old technology, but it has very much application in the recent days. Okay. So what is this monoclonal antibody technique? So we are using some experimental animals like rat, where it is injected with some antigens. Now, when this mice is injected with the antigens, then we know that the B cells, which will produces antibodies against this antigen. So, what we do is we isolate the B cells from the rat. Now, the B cell has a peculiarity, that is, it can survive in a medium where there is it can survive in a medium where there is a deficient de novo purine synthesis. Okay. Because B cells has one plus thing, that is it can survive with an alternative source of purine. What is the alternate source of purine? It is the salvage pathway, right? So the plus thing about B cell is that one, it has antibodies. Number two, that we are not concerned now. So here we should consider the plus thing about B cell is that it can survive in a medium where there is a deficient de novo purine synthesis, because it has the plus salvage pathway. It can survive with the salvage pathway of purines. Okay. Now we have a second set of cells, which is called myeloma cells. The myeloma cells, the plus about myeloma cell is it is a cancer cell. So the plus is that it has immense replicating potential. So myeloma cell has immense replicating potential, but the problem with the myeloma cell is, it has a deficient salvage, cannot survive in a medium where the salvage pathway is defective, because it is HGPRTase negative. Okay. Now we are bringing these two cells into a medium which is called as HAT medium. So what is HAT? HAT stands for hypoxanthine. H stands for hypoxanthine, A stands for aminopterin, and T stands for thymine. Okay, or thymidine. Okay, what is this hypoxanthine and thymidine? We know that or first of all, we should know what is this aminopterin. This aminopterin is a folate antagonist. Folate antagonist. So this aminopterin, which is a folate antagonist, always inhibit which pathway? Purine de novo synthesis. It inhibit de novo purine synthesis. Okay. So, we are making these two cells grow in a medium which is called as HAT medium. In the HAT medium, the problem is, or an unfavorable situation in the HAT medium is, it has no de novo purine synthesis. Okay. So, whatever cells has to survive in the HAT medium should have the alternate pathway of purine synthesis, and that is called as the HGPRTase activity. Okay. Now we have different cells in this medium. We have the B cells from the rat. Then we have the myeloma cells. Okay. So these two are growing in this HAT. Now in this medium, there is one more agent which is called as polyethylene glycol. What is polyethylene glycol? It's a fusing agent, which means in this medium what happens is, because of the action of this PEG, the myeloma cell is fusing with what? Myeloma cell is fusing with the B cells. Okay. Now, if they fuse, that is called as a hybridoma cell. Okay, when myeloma cell fuse with the B cell, that is called as a hybridoma. Why? Because it is a hybrid cell which has two important characteristics, that is myeloma cell has what is the plus? It has immense replicating potential. Now B cell, what is the plus? It can survive in a medium where there is no de novo purine synthesis. So this is an unfavorable situation, immense replicating potential. If it meets the what you can call as something which can survive in the Hat medium, that cell is called as a hybridoma cell. Okay, so that is it, where you can see these two fuses. You can see these two fused. These fused cell will survive in Hat, but the B cell that, or the unfused cell. Unfused cells are of two types. Myeloma cell will not survive because there is no HGPRTase. It cannot survive in that Hat medium because salvage pathway is deficient. What about B cells, unfused B cells? It also doesn't survive because it doesn't have immense replicating potential. So it won't survive. So these two will die, but what will survive is the hybridoma cell. Okay. Now, once it is fused, we know that these fused cells are, some cells will be having antibodies, some cells may not be having antibodies. So the next thing is that we have to choose those fused cells with antibodies. So that is marked red here. So that is a fused cell or a hybridoma cell with the required monoclonal antibody. Now these cells are, we are harvesting these cells. So we are harvesting these cells and we are cloning this harvested hybridoma cell with the antibody, and the myeloma cell, both are fused. So this hybridoma cells we are cloned either in vitro or in vivo. In vivo means we are again injecting it to a mice and it replicate inside the mice, so that we can harvest this monoclonal antibody again from that poor mice. Or other words, another option is like we can also do this multiplication or cloning of this antibody in vitro, that is outside anything like inside a test tube or something, where we can clone it, then also we can produce monoclonal antibody. Ultimately what we are getting is monoclonal antibody. Why we are so discussing much about this monoclonal antibody? There is no pharmacological agents nowadays without MAB, right? Everything is MAB. So what are the functions or what are the uses of this monoclonal antibody? Number one, it is in biochemistry and pathology, if you are saying, it is a diagnostic tool, because monoclonal antibodies will always bind to the particular antigen. So the sensitivity, that antigen antibody binding is highly sensitive. So this can act as a what you called as the diagnostic tool. For example, in ELISA, where antigen antibody interaction is used, then Western blot, where there also we can see antigen antibody interaction is used. So wherever this antigen antibody interaction, monoclonal antibody is highly specific, so we use that. Then in detection of cancer, because the if there is cancer, certain antigens are produced in response to the cancer cells. So if we can find out a monoclonal antibody against this antigen, definitely we can detect the cancer. Then serological identification is almost similar to the diagnostic application, nothing more to explain. Cell markers, certain monoclonal antigens and the monoclonal antibody, if it is binding, it helps as a cell marker. We can mark the cell using the antigen antibody interaction. That is again another thing. Then of course it's a research tool. Then another thing is like the treatment aspect. Treatment aspect is very, very important nowadays, because the patient tailored therapy we can do using monoclonal antibody. For example, in COVID, in the case of like almost every diseases which we can develop a monoclonal antibody against the antigen that is produced by these infectious agents. So again, therapeutic. And also the cancer, we can use it as a treatment of cancer or any other disorder where some antigens are produced, and if we can produce an antibody against, monoclonal antibody against this antigen, definitely it is a treatment. So which means that it's a research tool, it's a diagnostic tool, it is therapeutic. So that is why this monoclonal antibody technique, even if it's a very old, not, I won't say very old, but still it is not a very recent technique. Even then, this is important because monoclonal antibody has lots of applications. So going to the last question, which anticoagulant is present in the light blue Vacutainer used for estimation? Sodium fluoride, heparin, sodium citrate, or potassium oxalate. So the color coding of this Vacutainers or the collection tubes, it is very important, very commonly asked for the exam. This topic in detail is covered in the pathology session. That is why I have not covered it in biochemistry. So I just give you the different color coding just for revising. You all know the color coding. So first remember red. The red is something which there is no anticoagulant added, that is used for serological examination. That is majority in biochemistry. What we get is one, the red. The second that we are usually getting is the glucose. So glucose always remember it is the light gray color. What is the anticoagulant used there? Fluoride oxalate mixture. So you can see sodium fluoride plus you can also see oxalate. Then the rest is mostly going to the pathology part. So one is the EDTA. The EDTA mixture, especially for the hemogram, ESR, etc. This is almost having a light pink color. Then this is the question that is that was asked, that is light blue color. Light blue color is having an anticoagulant which is 3.2% sodium citrate. So this is usually used for coagulation studies like prothrombin time, then APTT, etc. So this is the light blue. So in that question they asked us the light blue. So the answer here is sodium citrate. Then what about this fluoride, heparin and oxalate? So what you can see here is the heparin. The next one is heparin. Heparin is usually that green color code is used for estimation of bone marrows. Like bone marrow studies, we use this heparin as an anticoagulant. Then comes another one. The citrate is present not only in the light blue, the citrate is also present in the yellow color coded collection tube or the Vacutainer as well. That is used for the blood culture. Then there is one more which is the EDTA itself, that is potassium EDTA, which is basically used for blood bank studies, ABO grouping, etc., mostly going for the transfusion medicine part. So that is almost a lavender colored bottle. So in biochemistry, what we are concerned more is the red and the light gray. These are the major one that we are getting. Apart from others, you can see that you can mostly going for the pathology lab, also to the transfusion, etc. All these are going. So remember, citrate is used in two color codes. One is in the light or the yellow color as well as the light blue. So the question here is very simple. So the anticoagulant light blue Vacutainer is citrate. Now what about the oxalate? The oxalate mixture you can see here, the sodium fluoride, potassium oxalate mixture you can see in the light gray color. So light gray. Then what about heparin? Heparin is seen in the green color code. Then the sodium fluoride, that is also in the light gray. So the answer here is sodium citrate. So that is about the INI-CET discussion, November 2023. As you have seen, almost all questions were very simple, except the one question that I give maximum difficulty level was the DNA repair gene. So for that also, if to explain, I need lot of time. So that is it. Apart from that, all the questions were simple, direct questions only. So all the best for you. Thank you.